He is the co-founder of Digital Garage. His name is Joi Ito. We are very glad to have him.
Thanks, Justin. So today's topic, as Hayasan, is microbes. And I'm not a microbiologist, but kind of like how we have lots of microbes in our body, I have lots of microbiologists in our network, and one of our biologists is here, and Sunanda will speak later. But for today, one of the faculty members and groups at the Media Lab that's been kind of immersed in microbiology can't be here today because he's in Napa at another conference that we're doing back-to-back. So I want to sort of focus a little bit on the way he's been thinking about it, Kevin Slavin, and sort of what his group is doing. And I think Sunanda will talk about the stuff from her group, Mediated Matter. So, and again, I'm going to try to set up the day a little bit too. So some of this may be somewhat redundant to many people, because I assume that if you're here, you already are kind of interested in microbes, so you sort of already know the story that, somewhere between, and the biologists can correct me if I'm wrong later, but somewhere between 50 to 90% of our body, depending on how you measure it, could be said to be non-human tissues, microbes, bacteria, viruses, and other things. There's probably like around, I think, 2 kilograms of microbial stuff in our gut. So you walk around and you think that you're a human being and that you're in charge. But in fact, we're probably much more appropriately characterized as a life support system for a microbial ecosystem. I mean, it's there's a word called holobiont, which Kevin likes to talk about, which is really when you think about things like human beings or any other thing, it's actually a system that kind of coexists with all the things inside of it. And if you think, another way to think about it is if you think about the fact that our gut stays roughly the same temperature, whether you're an Eskimo or whether you're in a hot desert. It's a very stable ecosystem, an environment for microbial life. And in fact, again, depending on how you measure it, our gut is probably one of the most sort of richest places for microbial life. And so if you were looking at human beings and larger animals as sort of spaceships where microbes could go where they couldn't go by themselves because they built a system that preserves temperature, feeds them, and somehow the microbes try to help us through the system. So if you think about it from the perspective of a microbe, we really are kind of like a spaceship. And I think it's also interesting, again, depending on the biologists you talk to, the microbes that we have, if you were to say, they're useful to us, so very necessary for life, and harmful to us, like some of the things like Ebola and things that will just kill you. Most of the microbes are somewhere in the middle. They do some things that are good for us, some things that are bad for us, and some microbes are really good and some microbes are really bad. But most of our history has been since we've discovered microbes, have been trying to eliminate the bad guys because they're the ones that make us sick. They're the pandemics, the epidemics. But in fact, in the process of doing that, we've eliminated and hurt a lot of the good microbes. So there are a lot of modern ailments, and we'll hear from speakers that are trying to reframe this conversation. But a lot of the modern ailments, like allergies and things like that, could often is said to be attributed to our combat against microbes or the hygiene revolution, which may or may not be, in the long run, a good thing. So one of the things, as we start to see how we've understood microbes, one of the ways we understand microbes is through gene sequencing. So there was a Human Genome Project, and it cost, I think, somewhere between $2 and $3 billion or so to sequence a human genome the first time. Now it's down to about $1,000 or less. And so as we started to try to discover more and more about the human body and got genome sequencing to get cheaper and cheaper and cheaper, it's gone faster than Moore's Law in terms of the drop in cost. And one of the best ways to understand microbes is to sequence their genomes, because then they're very difficult to see. And we're starting to identify the microbes, but we're still literally just a tiny drop in the bucket in terms of the total number of microbes that have been identified. We really don't don't understand this stuff. And what's sort of fascinating, and Justin mentioned fecal transplants, we have now discovered things that work, but we really don't understand how they work, because this whole microbial system is so complex that we are barely just getting to understand it. So we kind of got to this phase where we know enough to know that there's so much we don't know. And so as we start to do things like map the brain, we're starting to realize maybe that won't tell us how we think, because in fact maybe it's something else that's controlling our brain. But the one of the main instruments we're using is gene sequencing. When you start to sequence the genes, you start to realize just how many. And I mentioned that we've just scratched the surface. I think it's something like 9,000 or so bacteria out of maybe 10 to the 17th. This is a crazy small amount that we actually have been able to identify, even the ones that live inside us. This is an interesting graph. So these lines are different species of bacteria. And you can just sort of see almost all of the different things that we have been able to sequence. The eukaryotes here, that's like human beings and animals. It's a little tiny slice of all of the species on Earth are things that we, like Darwin would have put in his book. Most of life on Earth is bacterial. And so again, this is this interesting thing that we suddenly have this lens to look into this new world, and we're just starting to know how much we don't know. And I think this is exciting. But again, more broadly, when we think about science, which is really fascinating, is the minute we start to get to think like we kind of know what's going on, we realize we don't really know what's going on. This is one of my favorite stories, and this is one of Kevin's favorite stories. So this is a mouse that's been killed by a cat, and usually mice run away from cats, but in this case, this mouse ran and chased after the cat, or didn't run away from the cat, and it was killed by the cat. And the reason this mouse died was that it was infected by a microbe called Toxoplasma gondii. And with Toxoplasma gondii, it's an interesting microbe that only has sexual reproduction in the intestines of cats. And part of its life cycle requires mice it splits, and the mouse basically incubates one part of it, and then it has to then be eaten by a cat in order for it to complete its cycle. So what happens is when a mouse eats something and is infected by Toxoplasma gondii, the brain starts to behave differently, and it stops becoming afraid. So it's instinctively afraid of the smell of cats. But this microbe switches that off and makes it think that the smell of cat urine is a good smell, and it goes after cats. So this microbe is able to change the behavior of mice so that they do something that's not in their best interest in order for it to complete its biological cycle. Now, why this is important is that it's amazing that a microbe is able to remote, and there are many, many, many parasites that control things from ants to wasps, and it's kind of like remote control. They're able to get them to do all kinds of things. So when you start to study parasites, what you realize is, well, if these parasites are able to do this to mice, what's going on inside of us, right? So that would be obviously the next question. And so in the human being, we have this very sophisticated nervous system called the vagus nervous system. And we have tons of sensors all around our gut. And what's interesting is the neurotransmitters in our brain that our neurons use to communicate with each other, our microbes are already creating things like, and what's interesting is, these are theories, but there are some theories and hypotheses that say that many of these neurotransmitters may have originated from the microbes using to communicate with each other, then started to be used to communicate with us. And there's a lot of evidence to show that the microbes communicate with our brain through the vagus nervous system, and then our brain communicates with our gut. And that in Japanese we often say, hara de kangaeru, or think with your gut. and this whole sort of qigong thing about your gut being the center of your being, and it's very possible and probably likely that a lot of why we do things and how we do things has to do with the microbial balance and the communication inside of our gut. And so this is, again, just a lot of new research, but there is definitely some evidence. So it's clear that microbes affect the host's behavior. It affects it in many ways. A lot of things now, they're showing, even with the fecal transplants, that some of the neurological or psychological diseases and things may be transmitted, may be dampened, but it's a two-way system, because your brain can also affect the bacterial balance in your gut. So we're still trying to understand. It's a very complex system. And so this is a paper on how it may relate to anxiety and depression. And there's some really interesting things, because there's certain things, for instance, there are compounds in breast milk that the human body can't metabolize, but it turns out that compound feeds a microbe that then produces something that you need. There are some theories that say that maybe chocolate has some molecules that are very useful for your bacteria in your gut, and so the bacteria in your gut possibly are the ones that make you crave chocolate, that it's not your brain, but it's your microbial system. So it could be a lot of what we think about in terms of, and we'll hear more from the food people later, that your sense of taste and what you like could have a lot to do with what the microbes in your body like, and you just are sort of representing them as the starship human being that you are. And it could be affected by the microbes in your ecosystem. It could be affected by the microbes in your family, but it could also even be connected to some of our genetic code, because we are now learning that the epigenetics, that the code that you transmit to your children isn't just the DNA, but there's a lot of other meta-information that can be absorbed from the environment. So this whole simple image that we had of the brain and a genetic code that can't be influenced from parents to children, that's shattered now. And so we're just starting to understand that we're part of a much more complex system. So I think it's pretty clear that it's likely that our gut is a second brain. So we're trying to understand this new brain. There's another thing, which is, okay, the gut is inside of us. So, it's still part of, somebody described it in a really good way. Think of yourself as a donut. And the gut is actually part of the environment, because it comes in and goes out, but that's outside. So when you go outside, you see the city, right? So this is the Chris Mason lab at Columbia, New York, and what they did was they went around and collected the microbes in the city, and they drew maps of the city, of which parts of the city have what kind of microbes. It's really interesting, because you can see, for instance, even though it's been years since Hurricane Sandy, the places where Hurricane Sandy was, you can see microbes that have come in from the ocean, and different communities have different microbes. And there was a study that I think it was in Hong Kong, if I'm not mistaken, in the subways. You can literally see when people have come from certain provinces, because you see traces of microbes that are only in those provinces. So they showed that cities, people, and places have microbial sort of fingerprints. And so, and then Jessica Green, who's thinks about sort of architecture, sort of buildings and things like that. So she's done some really interesting work, for instance, with ventilation in hospitals. And one of the things that she showed, first of all, we spend a lot of time inside of buildings. So she thinks about the microbial systems inside of buildings. And what she showed was that if you think about sort of the likelihood of getting a pathogen or an infection in a hospital, her work is showing that if you close it off and sterilize it, you're possibly more likely to get infected than if you open the window, and probably more likely to get infected than if you were outside. And it turns out in a lot of the systems, even when we're trying to fix our own gut, that it's not about removing the bad guys as much as it is about increasing the good guys and balancing the system so that the community is healthier, rather than the elimination of the bad guys. The bad guys just turn into this very small, insignificant subculture rather than the rampant culture. Now, you can imagine for me, there are parallels. I did a session a few years ago at FUCAM called ISIS and the Microbiome, because one way, bombing countries and trying to get rid of the terrorists is very similar to bombing your gut with antibiotics. But what if you just made the community rich again? And what if you made the community vibrant again and diverse again? That may be a better way to push these dangerous subcultures aside rather than trying to eliminate them. So I don't want to stretch metaphors too much, but I do think that there is a parallel between sort of how communities and culture work and how microbial systems work. And so as we talk about food, as we talk about systems, I think we have a lot of intuition around how complex systems work in the way we think about culture, and that maybe we can apply some of those sensibilities to things like health. So but going around and scraping up microbes is kind of a crappy thing. And one of the things you literally do, one of the ideas is to instrument our sewage system with sensors to understand our microbial system. But being the Media Lab, we wanted to try to figure out what might be a little bit more elegant. And so what Kevin and his guys said is, how can we create microbial sensors for the environment? And they came up with this idea, what about the bees, right? Bees go around and get messed up in all kinds of things, but they come back home. And so what they did was they started putting beehives in Brooklyn, in Tokyo, and then what they do is they, well, first of all, a beehive has about a 2-mile range, and so you can, if you let the bees go out and they come back and you collect the debris, which is sort of the leftover stuff and the poop and things like that, you get a pretty good sample of the microbial environment that these bees have just interacted with. So what they do is they collect the debris and they sequence it, and they're able to see all of the different bacteria that are in that neighborhood. And so they've created a new kind of beehive that includes this little pan on the bottom where the debris collects. I'm going to skip that. And so they started putting these beehives all over different cities. So in New York, in Sydney, and this was in Melbourne. And they were at the Venice Biennale. Now, interestingly, the bees in Venice came back with a microbe that is a bacteria that is associated with rotting wood. And so they were able to show from the bacterial fingerprint that Venice has lots of rotting wood, which is not a good sign, obviously. But and then in Tokyo. And so what's interesting is that as they start to map the different bacteria that they're finding with where they are, and that they can see sort of what's going on in the environment. And what's interesting is every city has a different profile, right? And so that's, first of all, that's sort of interesting. And then what's also interesting is, this was the slide about the rotting wood. You can kind of tell by looking at the bacteria what's going on in the environment, because you wouldn't have lots of rotting wood in a place that was super dry. And so what can we learn by looking at these fingerprints? But what's also interesting, even just in Tokyo, you see that every neighborhood has a different fingerprint. So what does that mean? And so, I mean, we talk about things like sourdough, tastes good in San Francisco because of the microbes in the air. And I remember when I was in Shodoshima going to the soy sauce, I guess they're kura. The town has a different smell when you have a fermentation business. And so it's interesting. There's obviously intentional fermentation, but there's a bunch of microbial things going on. What does this mean? And then how does that affect culture, for instance, would be an interesting question. So, I think that we're going to see a lot of other presentations throughout the day, and it might be a little bit confusing on why we were talking about culture and food and things like that. But what I think is really important at the Media Lab for us is we often talk about anti-disciplinary work, which is working between and beyond the disciplines. And we often talk about the relationship between art, design, science, and engineering. And what's, I think, really key is when you're looking at a new science, which is so complex that we don't really understand it completely, where it is, but we can interact with it. That maybe some of the tools. So traditionally, what you do is you have some science, basic science, that creates some insights, that gives you some tools, that allows the engineers to render those tools into things. So for instance, if you think about the moonshot, you had the Newtonian laws that allowed you to understand basic physics, that allowed us to program and plan on how to get something off the Earth to the moon. You couldn't, or you wouldn't go to the moon using trial and error if you didn't know how, if you hadn't turned Newton's laws into engineering. But with the microbial system, it's kind of weird because a lot of the things, just like we're now realizing that Newton's laws, when you put them next to special relativity and general relativity, that they turn out to be local ordinances that are only true in certain cases. And now with gravitational waves, we're learning a lot of new physics. And then we're able to think about things like quantum computers. But weirdly, some people were able to build things that look like quantum computing before we really understood how they work. In the microbial system, we already have a lot of cultural understanding of microbial systems before we really understood them scientifically. And now with this understanding of the connection with the brain, finally the science is catching up to a lot of things that we sort of felt intuitively. So one of the things that I like to think about, and Alan Wallace, who's a famous physicist, but also he meditates, and he's a guy who, I think at Stanford it was, discovered that, or was able to prove that lucid dreaming happened in real time, because there's a theory that your dreams were just memories of things as if they were real time, but they didn't. There was no proof that memories ran in real time. But when he was sleeping, sorry, this is sort of a long story, but one of my favorite stories. So when he's sleeping, there was a person who had eyes were going back and forth, and they woke the person up and said, "What were you dreaming of?" And they said, "Oh, he was watching a tennis match or something." So they said, "Okay, well that's interesting." And so Alan Wallace set up an experiment. He was very good at lucid dreaming, so he went into a dream state, and they said, "Okay, when I'm in my dream, once I'm aware, I will send you a signal with my eyes." And so they waited, and then he sent a signal with his eyes, and he was able to communicate to his students from inside the dream using his eyes. But the reason he was able to do that was he was a practitioner of lucid dreaming, a practitioner of meditation, and he often talked about how you dig the tunnel from both ends. On the one end, you practice and you try to understand things intuitively to be able to ask the right questions in order to build the science, in order to build the engineering. And I feel like when we think about microbial systems, that may be one of the things that we should do. We should work with the artisans, the multi-generation homes that have been working with fermentation, have been interacting and communicating and collaborating with microbes, together with the scientists who have the instruments, who have the openness. And the interdisciplinary part is really that. I think you need to be able to develop tools, you need to be able to apply things like culture and design to science, and science back to things like culture and food. And that relationship's very important, and I think that's when you look at the sort of the success of different fields. For instance, I think video games are a really interesting field because the people who designed video games also created the tools. So they were able to shift business models from CDs to so from copyright to to online item sales and things like subscription models and things like that. And the games keep getting better, whereas other industries like newspapers or television, you see a real slowdown in the progress because the tool makers, the printing press people and the studio equipment people, are separated from the people who are making the content. So in a weird way, for me, and this is what we do at the Media Lab, we have people who are the designers and the artists also making the tools that give us the creativity. And I think this conference is very important because we have the practitioners together with the people who are doing the science, who are building the tools, so that we can think about these in a much more creative way, because the standard scientific process and the standard medical process, I don't think is going to make as much progress in trying to understand collaboration with microbes as if we were sort of working on this together. Thank you.